Oh my gosh, so many things to keep track of. Ah. Yeah, go ahead. I guess go ahead and do that too now. It's relatively straightforward. Associate number. Any other?
Yes.
Also? Yeah, I guess I'll do it now since that's fine.
So
I'm gonna have to pay for it on PayPal. So figure it out. Yeah, it's you email it to me. Oh, okay. After forward it to me, and I'll know which guy.
Walmart committed me.
It's 45. As well. All right. I'm going to make you modify your app that you played. Still waiting for Dr. Reynolds, so hand back your preparation homework. Your status is there. Birch. There.
Right here.
Oh. Hey, good job. Nice hit. Chris, copy. Kayla, grab it. What? Oh. So. Oh. Kayla Crowder. Jordan. Elizabeth Ellis. Sam Giddings. Taylor Goldberg. Caleb. And Jones. Anna Jones. Tyler Jorgensen. Kevin Kim. Kelsey Lanterman. Austin Maskovich. Good. Austin McIntyre. Connor. Asa. Cora. Kate Lindermeyer. Keith. Peter Cookjoy. Kennedy. All right, the key to this is posted on iTunes U, and the key to the practice room is also there. Although I should have put that because that's due at the end of the day. Oh, I forgot.
Dr. Kowinski, have you already asked whether the study session generated any more questions in their minds last night? He's doing something else. Did our study session spur any other questions for you?
I did not ask. Did it help at all?
It did help? Okay. That's good. The projector doesn't seem to be working right now. I'm going to turn it off and turn it back on, but while we're waiting for it to sync to itself and get turned off and turned back on, we'll just go ahead. All right, so what were some of the take-home messages from class? You don't remember, you can look back on your notes. Last time we had a class on this. Okay, so population growth, you said that you have to take into account birds. It does, right? And you have the equations for all the different. So what are the other, what are some of the other variables? So population size. What are some of the other? So, length of time, it could be years, it could be minutes, depending on the species.
Well, now it's working, but the screen's not down.
Right, it's because I told it all to turn completely off, and it decided to turn off the screen, but not the.
Pay no attention to the map.
Okay, all right. So, what else? What else do we put into fitness? Okay. And does it have a symbol? You are. What else can you tell me about the population growth equation? Little BT is the birth per capita.
Sorry, I wasn't paying attention. What does that fitness as R mean?
Someone said there's fitness, and I said, what is the representative of fitness in the equation? And they said little r. How do you feel about that?
Little r wasn't it the instantaneous growth rate?
Okay, so it reflects the fitness, perhaps. But is this an individual measure?
That was actually going to be my concern.
Okay.
It expresses how many theoretical offspring are created in a given instance.
By the population as a whole, per individual?
Population as a whole.
Okay, by the population as a whole. So perhaps let's not think of that as fitness. We're going to think of this as population growth rate. Is there anything you want to add to this or lead them to, Dr. Klinski, as far as little r?
No, it's in the continuous time frame, but my
So
question about,
instantaneous?
yeah, it's instantaneous. So my question was basically, R is a characteristic of population, and I'm at least in the habit of thinking of fitness as being measured at the level of the individual.
I would agree with that.
That was my own. But I wasn't listening to that thing to the screen.
That's okay, thank you for
I
your
looked up there and I'm like, whoa, what is that?
Wait, what does that mean? Okay, so I'm going to actually stop here. There are other things about this equation, like the actual equation, right? There's what is lambda, there's how you put all those together, how you calculate them. We do have a lot to cover today. If I said, hey, what did we cover last time? What can you tell me about population growth rate? And you struggled, make sure you spend some time on that this weekend, because not only will you have to look at the equation and then plug things in, you're going to have to know really intimately what each symbol means and why it goes into the equation that way. If you know that, then you'll be able to use the equation well, given whatever circumstances in the problem you have to solve.
Preparation homework, if you didn't do the calculations correctly, I wrote check the key. The key is on iTunes U. Hopefully it makes more sense to you now than it did before you had lecture on Wednesday. And at the end of the day today, I will post the key to the practice homework on iTunes U. I did it earlier today and I forgot it's not doing 5 sites of the town, but I'll put it back up and then you'll have that to study from.
The population growth homework is due at 5.
So if you want to take yours back, I can give it back to you.
If you've already turned it into Dr. Klinski, you're all set. Okay. So today we're going to shift gears a little bit and kind of go back to thinking about genetics. Today we're going to focus on the central dogma and mutations. And the basics of this is that mutations are the source of all heritable genetic variation that exists ever. It is the source. And the central dogma is an explanation of how the information in your DNA gets started toward becoming expressed as your phenotype. And we'll go into that in detail as we go through. All right, so the central dogma is this flow of biological information, right? And that flow is one direction in most eukaryotes most of the time. DNA stores that information in the nucleus. When your cell gets a signal that it should make some proteins, it copies that DNA message into RNA. And that copy of RNA can leave the nucleus and go to that protein creation machinery and get translated by that machinery. That message gets translated into a physical protein. And this is an overview, and we're going to go into each of these steps at least once, more like 3 times throughout today. And I'll post these slides. Okay. So central dogma is this portion of the process from DNA information to organismal phenotype. It's this first portion, DNA to RNA to protein. So what I would like you to do is to draw a concept map of that process in your notes I know it's easy to think through that concept map or visualize what it would look like in your head. I do want you to physically draw it in your notes. And once you draw a concept map of the central dogma, I want you to take turns explaining it to each other in as much detail as you can. Now, I know that you guys know a little bit about this stuff already because most of you had biology either in 8th grade or in high school, and you may remember parts of how DNA gets translated into protein. So once you have your concept map, then turn to a classmate, show them your map, and explain it to them as if they've never heard of it before. Okay guys, I see you guys are kind of pausing and looking up. Turn to each other, walk each other through it, take turns. Even if one of you explains it, that doesn't finish the exercise. Then another person has to explain it. Your concept map itself doesn't have to have a ton of detail. Just when you're explaining it to the other person, add as much detail as you can. Verbally.
We start with mine.
Just so you know, the reason she's asking you to do this is you learn the things you're trying to learn as you're trying to explain it to somebody.
So
There's no
DNA
other reason.
sends info into the nucleus. That info from the DNA sends out RNA for copy, or sends out, yeah, so the DNA sends out the information of RNA of a copy to the cytoplasm. The proteins are then made, and the ribosome translates the RNA message and it creates proteins.
Yeah, so we have DNA is in the nucleus, and it's transcripted in the cytoplasm into RNA, and then RNA is translated by the ribosome into a protein that's made up from the amino acids. I just take this and see this is really
Yeah, also this recording.
it should give a cell molecular answer to this on a DNA test. Is that okay? Because it's probably what's going to happen.
As long as you actually answer the question. Okay, then you're good. Go ahead and explain the difference Question? Okay, all right, let's come back together as a class. All right, so hopefully you have a concept now, maybe your notes now, you can refer back to and you talk to each other, and so you have sort of this base understanding of what's going on with DNA being the information storage molecule, RNA being the information transfer molecule, and the protein being the result. All right, so this is just a concept map of this flow of biological information in the cell. I've got DNA up there, and then there's transcription. So to transcribe means to write something down, right? So this message is copied into RNA. We're going to mostly talk about mRNA today because mRNA is what's made into protein. There are other types of RNA. I will not ask you that in the exam. I will ask you that next year. There's ribosomal RNA, there's tRNA, there's dozens of other kinds of RNA. Really the one that goes on to get translated by the ribosome into protein is that mRNA. On an exam, if you say RNA, at this point, that's fine. Okay. So if I were to ask you for a definition on an exam, for definition of the central dogma, I would want that it's the flow of biological information, but I would also want the specifics, that it is from DNA to RNA to protein, and that it's unidirectional. We never have a situation in the cell, to our knowledge, where protein is back translated into a nucleic acid message. It doesn't happen. So as far as making proteins, it's always that direction. DNA, RNA, protein. There are some situations where RNA can be translated back to speech. Mostly by viruses, but also within our cells. But yes, which is exciting. We'll talk about that next year. Okay, so the top part represents transcription from DNA into RNA. The bottom part represents when the RNA then leaves the cell, or sorry, leaves the nucleus, stays within the cell, leaves the nucleus where the DNA is, and goes to the cytoplasm where the ribosomes are, and they are those little protein factories, the ribosomes. All right, so we're gonna go over this a slightly different way. Rather than looking at it in words or concept map, we're gonna look at a cartoon of the molecules. So here we have a ridiculously short piece of DNA. We're going to pretend that genes are ever this short. Here we've got a cartoon of a structured double-stranded DNA. So each of these blue lines represents the sugar phosphate backbone of the DNA. Each of the colorful rectangles represents the nitrogenous base that's attached to those nucleotides. The purple blocks along the top are a prediction of which nucleotides will be grouped together to be translated into amino acids much later down the road. So for right now, we can ignore those purple blocks. Right? So the point is we've got this double-stranded our DNA that we're starting with. This is the message in the cell. Now, where is the DNA? In the cell. In the nucleus, right? Okay. So the first thing that happens when we want to make protein is that DNA, the hydrogen bonds, non-covalent bonds that are between the nitrogenous bases on two separate single strands, those hydrogen bonds are destabilized so that each strand comes apart. So now we've got this little transcription bubble. We've got this place along the big strand of DNA where those strands are separated. That allows some proteins to get in there and use one of those strands as a template for making RNA. That's transcription. A protein comes along and uses this and says, okay, I'm going to make a strand that can hydrogen bond to the nucleotide of this strand in this order. And you'll notice that the sequence of the non-template strands and the RNA is relatively similar. The one difference is that they're used in RNA 15. I won't ask you that on an exam this year. What I will ask you, what you should know, is that the DNA separates, and one of the strands is used as a template for making RNA. Now, this is all occurring in the nucleus. After that RNA is made, those strands of DNA hydrogen bond back together, just existing stably. Now, what do we need to do with that RNA in order to turn it into protein? What's the first step?
Get it out of that nucleus.
Get it out of that nucleus. There are no ribosomes inside the nucleus. You can't make protein inside the nucleus. So it's transported out of the nucleus into the cytoplasm. Cytoplasm is basically water and a bunch of other molecules and organelles that are inside the cell. And at that point, this message is a coded message. It has some words, the way that we have words in our languages, that are then translated by the ribosome into a protein. And the way that it's translated is each triplet, each 3 nucleotides is a separate word. And each of those words means a particular amino acid. Proteins are just strings of many amino acids. That's all they are. Loads of amino acids in a particular order. And so because there are codes for specific amino acids in a specific order, then we get this nice linear protein with amino acids in that order. The way that happens is that a little adapter tRNA molecule comes in and brings in the amino acids to the ribosome. I will not ask you about tRNA or about the molecular mechanisms of how this happens. But again, what it's using is it's got some nucleotides here that are complementary to the nucleotides in the mRNA message. UAC, if it were DNA, would be TAC, right? So it's basically just using that nucleotide complementarity to bring in the appropriate amino acids. And then each of these is covalently bound to each other. So in the end, we have this linear message of DNA that gets transcribed into a linear message of RNA. And that linear message of RNA gets translated into a linear sequence of amino acids, which is your protein. This is just an example. This is the codon table. Most organisms on the planet use the same code for which triplet of nucleotides gives which amino acid. Most organisms on the planet, not all, but most. I just wanted, that's just an example. And then in the end, we get this long linear protein, right? Here's our example. In the context of this protein, which is 268 amino acids long, this is just 7 amino acids, so it's just tiny. This tiny segment right here. Okay? But we end up getting this long linear protein. Then, this protein, like all other molecules in the cell, has chemical characteristics. What are some of the chemical characteristics that you guys know? Can you think back to chemistry? What are ways that you can describe a molecule or a compound? Asa?
The protein contain minus.
Say it again.
The proteins contain a group of components of amides.
Oh yes, yeah, absolutely. So protein contains amides. So amides, you can sort of take the am part. It refers to amino groups, amino acids. Sure, yeah, you could describe them in that way. What about even more general, like if somebody gave you any molecule or any chemical, what types of things could you use to describe it?
Polarity.
Polarity, absolutely, polarity, okay? So if it's polar, it's gonna like water. If it's non-polar, it's gonna be like oil, right? And it's gonna seek out other oily sections and stick to them, and the polar parts are gonna seek out other polar or charged parts that, to stick to them. Because of that polarity and charge, this linear protein, like all proteins start out, is going to fold into a three-dimensional shape. So this protein here, 268 amino acids long, I actually looked up on the protein database. I don't know. Thought I did that, but I didn't. Well, I'll show it to you here. Protein database, you can actually look at the three-dimensional structure of these guys. I don't know if you can see it very well, but in this picture, you've got some sections that remain relatively linear, and then you've got other sections that become a helix, kind of like DNA does. It's an alpha helix. You've got some sections that are represented by these flat arrows, and it's because they folded in layers like this to make a sheet, right? So basically you have this linear protein that then folds up and makes a three-dimensional molecule within the cell. And that three-dimensional molecule can then go and do jobs in the cell based on its shape and its charge. Okay. So that's like kind of the mechanics of the central dogma, how you get from DNA to a finished protein. Now, there's some other details about, some of the proteins get modified along the way, but as far as the basics, that's that. Okay, so what I'd like you to do now is, again, turn to a partner and explain this process to them, now that we've talked about it in a little bit more detail. How do you end up with a three-dimensional protein?
Yeah. So separated
So then once we have like amino acids like that,
I just really like everything you can and then because similarity of the amino acids to itself
would be like a
or
magnet,
whatever.
right? So you've got the negative together, it's gonna stick. Unless it sticks to the proteins, they have to be in a structure, it has to be one of the so it has, you don't want one enzyme, one change its nucleus.
No.
And there's really only.
Weird class.
And because you have so many different ones.
There's like a stock this year, we had just like
We did this like salad, and like it was like barley, it was like circles, like it was.
we had to make a chart of like these parts. I think I still have that.
Okay, did this bring up any questions? You guys have questions for me?
Yeah, it's like a chart, like
You do have a question. Okay.
I don't think we can right now.
Oh, yeah. In the transcription part, it's only mRNA. All right, okay, so now we've talked about the central dogma, but that doesn't tell us how our genes lead to phenotypes, right? Because at this point in this class, we've talked about the plumage color on coots. We've talked about horn length in lizards. We've talked about beak size and shape in finches. We've not talked about the RNA and the protein and what's going on there. I mean, we know that the gene and which form of the gene that you have leads to the phenotype, but there's kind of a big black box in there, right? Okay, so the first part of that is, what does the gene do? It gets transcribed and then translated into protein. Some genes do not get turned into protein. Some genes make RNA, and RNA goes and does a different job. We'll learn more about that next year. But the best examples for this to help us understand how genes become phenotypes is to look at these protein coding genes. We are going to talk about how the protein then leads to cell function and how that cell function gets turned into that specific phenotype. But before we talk about that, I want to talk about mutations. Do you have a question, Asa?
Yes. There are a lot of theories that there's a lesser amount of DNA in the mitochondria of the cell. Is that true? And if it's true, does always genes undergo transcription?
Yes. So let me make sure I know your question. So you're saying, is there less DNA in the mitochondria than in the
Mitochondria, some amount of DNA.
They do, they do. So what we understand is that long ago, before there were any eukaryotes, a bacterium was engulfed either by another bacterium or by an archaean. There are three main taxa of life: archaea, bacteria, eukaryotes. Eukaryotes are what we are. They can be multicellular organisms. They have lots of organelles. Bacteria can sometimes have organelles, but typically do not. They don't have nucleuses. Archaea look, under the microscope, identical to bacteria, but their molecules are super different and look much more like ours do, right? Okay, so mitochondria, we think, was a bacterium that was engulfed by an archaean and became the first organelle. It actually still retains its own genome. So you have ancient bacterial DNA in every single one of your cells, all of them. So it has its own circular genome in there. It is smaller than the genome of us, in the nucleus, quite a bit smaller, but depends on what organism you are, how much smaller. Does it undergo transcription and translation? There are no mitochondria in the nucleus. Yes, it undergoes transcription and translation. It does some of its own inside the mitochondria, and some of it is actually now bits of mitochondrial DNA have been sort of accidentally taken up into our nucleus And so we transcribe and translate the rest of the cell. The nuclear genome transcribes and translates proteins that then are transported into mitochondria. Mitochondria are cool. Chloroplasts, we think arose in the same way, and they do a lot of the same kinds of things.
I don't know that you want to mention this, but it's also cool that they're only inherited maternally, so they're inherited genomic DNA, and that has all sorts of implications for like recreating the phylogenies of organisms, and they are pretty cool.
Yeah, they're really cool. And mitochondria are maternally inherited in most eukaryotes. I don't know anywhere that have, there is some paternal mitochondria that leak in sometimes, but it's typically above 90% just from mom, because those mitochondria are in the cytoplasm of the oocyte that gets fertilized. Chloroplasts can be, it depends on the species, whether they're inherited maternally or paternally. But this is why often they'll talk about, evolutionary Eve, right? The mother of all. Right, mitochondrial Eve. It's because we can track the human evolutionary history by looking at the DNA in the mitochondria. So that's why they call it Eve instead of Adam, because they'd call it Adam if they could. Let's just be honest.
Back in the old days, we first did this with mitochondrial DNA because it was smaller and simpler and easy to access, and that was all we could do at the time. So these early reconstructions of human history were done only with mitochondrial DNA.
Well, it turns out mitochondrial DNA changes very, very slowly.
It also does very, very slowly, so you can look at deeper evolution than you can more recently.
Right. If you have something that changes really fast, you can track it to a certain point in history, and then after that, you're like, I don't know what the hell happened before that. Could have been anything, really, because it's so different in a rapid period of time. But mitochondria stays mostly the same. Okay. So now we're going to talk about mutations being the source of all genetic variation, and we'll talk about effects of mutations. That'll lead us back to how DNA contributes to cell function, and then causes the organismal phenotype. Okay, so mutation is the source of all genetic variation. You do not have a genetic variant that did not arise by mutation. Think about that for a minute. I'm going to ask you to ignore these other things that you can think about. You do not have, there's no genetic variation that didn't come from a mutation. When you normally think about mutations, what do you think? What does that bring to mind?
That's like a really bad connotation, I feel like.
Yeah, it has a bad connotation. Okay, and tell me more. Somebody tell me more about that. What else does it kind of hint at?
Usually, like when people think of mutation, they think of like birth defects or an abnormality, so something that causes something wrong with your DNA, but that doesn't necessarily always do that. Right,
yeah, absolutely. So when you think mutation, you think, oh, something that's causing dysfunction, something that's causing something really wrong.
Well, I usually just think of the more mundane, random mutations that distinguish such as melanin deficiency in Homo sapiens originating on
There you go. There you go. Okay, and so when you start thinking about genetic variation, you have to then allow for the fact that some of these mutations are not going to be harmful. In fact, the majority of all mutations that occur are neutral. They have no effect whatsoever. And some of them that occur are beneficial.
lactase.
Like lactase. Helps you digest milk even as an adult, right?
But not in the case of cancer.
What was that?
But not in the case of cancer.
Right. Or lots of us who are, lactase deficient, right? Okay, so mutations are the source of all genetic variation. So then that brings up, okay, well, what are mutations caused by? And this was the first bullet point in the thing that I erased. So don't be too mad at me, because we'll fill it in. Causes of mutation, physical stress on DNA or chemical stress on DNA. What are some things that you know of that would cause physical stress or chemical stress on DNA?
UVC.
UV radiation, right? This is why you want to wear sunscreen most of the time if you're going to be outside for more than 15 minutes, because UV radiation will cause physical stress on your DNA, because that radiation will actually penetrate your cells and cause mostly the adenine and thymine in your DNA that are in separate rungs of that spiral staircase to actually link to each other. And when they do that, and then your cell tries to replicate its DNA, it will not work. It will either cause a deletion or it will cause the incorporation of the wrong nucleotide complementary to that. So UV radiation, what else do we know of that would cause physical or chemical stress on DNA? Asbestos? Yeah, absolutely. Yeah, that's a chemical that will get inside your cell and mutate your DNA. Yeah, it'll cause chemical stress. What else do we know? Carcinogens, yeah, smoking. Carcinogens come from, carcinogen really is something that is defined by being cancer causing, right? So when you get mutations, that can lead to cancer. Those are bad mutations, right? Okay.
Do viruses often cause mutation?
They do, and in fact, they cause some cancers. Some cancers are only caused by viruses.
Laser radiations.
Pardon?
As well as laser radiations. Laser radiations.
Yes, yeah. So anything that's going to come in and interact with your DNA can potentially cause mutations.
How about mistake by transcriptase?
Absolutely. So anything that comes in and interacts with your DNA, even stuff from your own cell. Oh yes, it has been shown. Even when your cell is wanting to replicate itself, it's interacting with the DNA. It can make a mistake. It can actually accidentally damage itself. It can sometimes will purposely damage itself in order to accomplish something else, but then it will imperfectly repair it, right? So anything that interacts with your DNA.
This is like a cystic fibrosis.
They're certainly caused by genetic mutation, absolutely. Yeah, so you can get diseases that are caused by it. You can have a neutral, or you can have something harmful, which leads to the next thing that I want to talk about, which is the molecular effects. Like when I talk about a mutation, what the heck do I mean? Most of the mutations that we are going to focus on in this class are mutations that are maintained at some level within the population. They become alleles. Okay, so an allele is a mutation that sticks around in the population, becomes a common thing. So an allele is one form of a gene. It was a mutation that occurred, and is now relatively common in the population. And when I say relatively common, I mean anything above something like 3%. Okay. Frequency.
So the what's it called? Royal disease thing is not an allele in that definition.
Oh, and yeah.
One
Not
that,
in India.
Satyavich, I say that.
I know exactly what you're talking about, not my brain is not coming to the name. Hemophilia. So,
Specifically, it was passed on from generation to generation in this archetype.
yeah, so within that, because that was a closed breeding population, it was probably higher than 3% within that closed breeding population.
So it's a proper relief.
So it is a proper allele, absolutely, absolutely. Right. So now we're going to look at, we're going to have this little short fake gene here that's not really as long as the real gene would be. Real gene is somewhere between 2,000 and 20,000 nucleotides in length. This one is 12. Okay, so we're just going to pretend that this is the wild type version. So when we say wild type, we mean sort of the common, the most common form in the population. Wild type. Most common. Okay. It may not even be the best, right? You might have a beneficial allele out there that hasn't become most common yet because it's either not being acted upon by natural selection or has a more complicated situation. But this is the wild type allele. It's most common. An allele is defined by that sequence of nucleotides. We could have a point mutation. Okay, so let's say this UV radiation comes in. It makes an A and a T can't think of the word, makes them bind together inappropriately. And then during the next round of DNA replication, your cellular machinery reads that weird AT as actually being a C. Okay. And there's not actually an AT here, it's AG, but the A gets changed to a C. So we can have a point mutation where one nucleotide is replaced by another. So the UV example is very good. My apologies. In this case, maybe it was a carcinogen or asbestos or something. Something damages the DNA, we get a point mutation. One nucleotide changes. Now you can see these purple bubbles above here. These represent the amino acid that would eventually be coded for by that triplet, that codon, which is 3 nucleotides. So for example, ACA is going to turn into, it's going to be eventually translated into threonine, the amino acid threonine. Compare these two from the wild type to the point mutation. Will these two strands of DNA be transcribed and then translated into the same protein? Yeah, it's identical. One of the tricky things is that sometimes, for some amino acids, it doesn't matter what the third nucleotide is. So this is why many mutations, most mutations are neutral. It's because they either don't change the amino acid at all, like in this case, or they change an amino acid that's not very important within the function of that protein. But in this case, this is a silent mutation. It's mutated to a different nucleotide, but it's not translated to a different amino acid. We could have an insertion. We could have an extra nucleotide or more than one extra nucleotide inserted. And when that happens, particularly if it's not a unit of 3 that's added, it's going to have different consequences. What can you see about this mutation, adding a G between these two A's? How does that change the protein?
The G replaces A.
Pardon?
The G replaces A.
It doesn't replace A. It goes in between the two A's in this example. It's an insertion.
So the entire G is shifted, so we get a whole new thing in it. If it's at the beginning of the G, especially harmful.
Right. So now we've shifted that 3 nucleotide code such that our triplets are completely different. So ACA, that's maintained, but An AAG, which would make lysine, is no longer there because we have we have GAA, which makes a totally different amino acid. So this is going to lead to a protein that has protein, and then the rest of it is completely different. Different charges, different hydrophobicity. It's going to fold into a different shape. What do you think is going to happen to the function of that protein in the cell? It's not denatured. Denatured means that it's going to not be in a three-dimensional shape anymore. Pardon? It's certainly not going to be functional because it's not going to bind with the appropriate binding partners. So it's not going to have the right function in the cell anymore. We can also have a deletion. Okay, so in this example, the A that was here, okay, so we got ACA, AA, one of those AAs was removed. So we still have ACA, but instead of then AAG, we have AGA.
So under deletion, is it possible for the portion of the amino acids to delete to just as an last rather than being replaced?
So are you asking, is it possible for
Chunk.
a unit of 3, a chunk that's a unit of 3 to be taken out? Yes. And in fact, that's true of insertions as well. So if you have a unit of 3 that's inserted in just the right place, then you might just have extra amino acids. If you have a unit of 3 that is deleted in just the right place, then you might have just a shorter protein, which may or may not be functional. So you can have this variation in protein effects. I'm going to go back for a moment to this substitution up here. With this substitution, in this example, we substituted that instead of an A here, it got mutated and it was a C. And because it was in that third nucleotide position, it didn't really change which thing was being coded for. In this example, the nucleotide that was mutated was one nucleotide further upstream, and so it changed AGA to AAA, which then did change the amino acid. Sometimes the change of one amino acid can change the protein so much that it either has no remaining original function, or it has a totally new function. It can be quite drastic. It can be neutral. It depends on where in the protein that amino acid is. I am not going to ask you about under which circumstances would a substitution be have an effect on the protein or not. Okay? These examples that I'm giving you are to help you understand what do I mean when I say mutation. So you can have a substitution, an insertion, or a deletion. And to understand the range of potential effects on the protein then. It could change the protein sequence. It could truncate the protein, make it way shorter by introducing a stop message. TAG means don't translate after this. So it can either replace amino acids, you can take them out, you can add them, and that's going to change the shape and possibly the charge of the resulting protein. All right, turn to each other, talk about this. Walk through what kinds of mutations can you have. Okay, I want you to tell me 3 general classes of mutation. I want you to give me 3 possible effects of the mutation on the protein. I'm going to hide this for a moment.
Three-channel class, which is insertion, deletion, and substitution.
So like insertion, add nucleotides with half of the protein and deletion so it can make it non-functional.
Okay, and then what are the possible
My favorite is truncation. Yeah.
Good deal. All right. Okay, sounds like you guys are on the right track here. I've got a pretty good understanding of what's going on. Okay, so you can have the resulting no change to the protein. It can change the structure of the protein. It can, change the acid. It can shorten it. It can lengthen it. One thing we haven't talked about is that it can change the amount of a protein that gets made. So if the mutation occurs in a regulatory region surrounding the protein coding gene, it can sometimes just mean that less of the functional protein is made. There's no dysfunctional protein made, just less or more. And having less or more of a protein can have either no effect on the organism or a good effect or a bad effect pertaining to fitness. So these are sort of the three types of impacts that can happen on the protein. No change, change in structure, or change in amount. Yes.
So if it does not change the first one, second, of course, no change the protein, in which case it will be substitution, right?
Right. It would be a substitution, right? It would have to be a substitution if it doesn't change the protein at all.
Now, mutations hold that there's always a change. So can we say it's a mutation?
Yes, it is still a mutation because it changes the DNA. Okay, but the resulting protein is not what we would call a mutant protein because it doesn't change the protein. So it depends on what you're talking about, whether you're talking about the DNA or talking about the protein. And in fact, you can have a change of the protein, but not see any phenotypic result. And so you would call that, say that the organism, there's no organismal phenotype, there's or mutation, right? It's not that the individual is a mutant, if they're not. But they do have a mutation in their DNA, possibly even in their protein. So it depends on what level of organization you're talking about. All right, so now we're going to talk about, in our last 5 minutes or so, we're going to talk about after you get the protein. How does that protein then impact cell function, and how does that cell function impact the organism, and then how does that impact phenotype? Now, it's a lot to talk about. So, but I think now that we've talked about these general mechanisms, that the example will go relatively. This is human chromosome 7. You can see there are a lot of color, black and white bands on the chromosome. The black and white bands correspond to either more condensed or less condensed DNA. They do not necessarily correspond to genes. But there is a gene inside here in the little red band, and the gene is called CFTR. And if we were to use our pretend fake eyeball microscopes, we might be able to zoom in on this compacted chromosome and uncompact it a little bit, and unpack it a little bit more, and unpack it a little bit more, and more, until we finally get down to the double helix. Okay. Then we adjust our eyeball microscope a little bit more, and we look in that double helix, and we would be able to draw a cartoon of the CFTR gene. Now, this cartoon representation shows you boxes where there are important functional parts of that gene. The areas between the boxes are regulatory regions and introns that either we don't know what function they serve because they're cut out before they're made into protein, or bottom line, those areas are not made into, they don't end up in the protein. Okay, so the boxes are the parts that get translated into the protein in the end. There are some parts of the gene that are not, but the boxes are. So from this cartoon, we can say, okay, well, if we just list the boxes, we take out those little in-between pieces, nucleotide, then we get this mRNA for the CFTR gene. And this the CFTR gene is typically about 6,500 nucleotides long. Okay, so it's 6,500. This mRNA is transported out into the cytoplasm and made into the CFTR protein. Now you can see that it has these kind of functional blocks here at the top. It has these sheets here, and it has a channel that goes through the center. And that's important because CFTR gene codes for CFTR protein, and that protein does make a channel. That channel goes through the cell membrane, okay? So here's outside of the cell, here's inside the cell. I apologize, the best and legally freely used available image of this particular protein embedded in the cell membrane happens to be in German. So don't worry about these words that seem to not mean anything to you because they're in German. So this is the protein itself, and it makes a channel through which chloride ions need to be pumped in one direction or another. Okay? This molecule determines the cell's ability to regulate how much chloride is inside and outside of the cell. Now, chloride is a salt. Okay? If you imagine a container with a membrane here, and it's filled with water, and you have lots of salt on this side, you put in a bunch of salt, and you have no salt on that side. And water can go through this membrane. Think about diffusion. What do you guys know about diffusion? Where's the water going to go? To the side with the salt. If this membrane is partially watertight, but not entirely, that water will move over to the side with lots of salt. It'll do that because the law of diffusion says that if you've got a membrane and there's various different concentrations of solutes, the water will go toward the highest solute, such that the concentration of solute is equalized on both sides. Okay. So we've got this thing, the CFTR protein, that is purposely putting a bunch of chloride ions out of the cell. It's like pumping them out. Okay, we have too many in here, right? And it is maintaining osmotic balance. When the CFTR gene is mutated, it causes cystic fibrosis. When it is mutated, this channel can no longer pump those chloride ions out. Okay, this is the normal. This is the channel, and it has this big blob of protein that opens up so the chloride ions can go out. Okay, and this opens in response to energy and in response to ATP. Okay, when it is mutated, it can't open anymore, and those chloride ions don't go out. When you have too many chloride ions inside and not enough outside of the cell, what's going to happen to the water in your body? Where's it going to go? Is it going to go toward the direction of lower salts or higher salts? Right, it's going to go into the cell in an attempt to dilute the salt concentration in the cell. It's going to mean the outside of the cell is super dry. Okay, so here's how this molecular mutation has now impacted the cell. Now, because the cell has loads of salts inside and the outside is very dry, okay, we're going to look at organismal effects. This will cause anywhere where you have tubes in your body, your lungs, your intestines, your pancreas, where it's going to excrete insulin. Those tubes are going to be super dry mucus, and they're going to get clogged. So at an organismal level, you now have clogged tubes everywhere. Give me a tube in the body. Well, okay, I have to restate this. You're not going to have clogged arteries or blood vessels from this. Non-circulatory tubes. Esophagus. You're going to have thick mucus that cannot be moved around to get all of the gross stuff that you hang on. I'm not a medical person. Trachea, esophagus. Trachea is breathing. Esophagus. Wait, what?
You're thinking trachea.
It would be true.
Right. What's your esophagus is where you eat?
Yeah.
Okay. Okay. So you can't swallow and get the food down very well. Give me another tube.
Trachea.
Trachea. You cannot cough out all the gross infectious particles that you breathe in throughout the day, so you get an infection. Okay. So you can think about this with any tube in the body. So bile ducts. You cannot release the digestive enzymes that you need to digest your food. Intestine. Your mucus in your intestine is so thick that what food you do digest, you can't take up. Okay. Your poops get really ginormous because you can't absorb them, right? Or you can't uptake the nutrients that you need to uptake. All right, so then we get fitness effects. What do you suppose the fitness effects of not being able to use the tubes in your body are? Do you think it'll affect survival? Yes, big time. Individuals with cystic fibrosis, even with all of our medical interventions, die usually by the ages of 35. What about fertility? If you live, do you think it would affect fertility? Yes, because you can't have sperm. We're past time, so we need to go. But if you have questions, let me know.
You have the homework on the table?
Yeah, leave your homework. You can hand it to me or leave it on your table.
Mistakes that had mentioned.
Oh, okay, go ahead and give the practice homework to Dr. Monkey. Practice homework. Yeah.
A meeting with research student like that, and I about 3. We have questions. Oh, yeah. Shove it under my body. And make sure that I just
Oh, you can download the application from the federal website.
And this is Travis. I need a copy of your, go online, print it out. Email. Yeah, sure. I'll deal with it Monday. Yeah. All right, cool.
I'm so angry with this So far, that's my favorite class example.
Cool beans.
It's my sorority.
I'm gonna post a little later. I did make sure to get permission from my friend, but my friends have a son with cystic fibrosis, and she does a blog.
Oh, cool.
And so it's kind of, she'll post weekly, and it's like, okay, sick again, okay, sick again, okay, sick again.